Good evening, everyone, and welcome to tonight's Science at Melbourne Public Lecture. My name is Professor Moira O'Brien, and I have the distinct privilege of being the Dean of Science here at the University of Melbourne. I would first like to acknowledge the traditional owners of the lands we are on this evening, the Wurundjeri Woi-wurrung people. I also acknowledge the traditional owners and First Nations people of the lands from which our online audience is joining us tonight. Aboriginal and Torres Strait Islander people are Australia's first scientists with deep and enduring knowledge of the land, waters and skies. And I pay my respects to elders past and present, and I particularly welcome any First Nations people joining us here tonight. The annual Science at Melbourne Dean's Lecture Series is an opportunity to share our faculty's knowledge, application and love of science with the wider community. Tonight's discussion, Electrifying Medicine, Revolutionary Health Solutions Emerging from Other Fields, is our fourth and final lecture of the year, and I anticipate it will be a fascinating one. And we are incredibly proud of these series of events, and it is a staple of our calendar every year. Public lectures have a long-standing tradition in higher education institutes all over the world, and for very, very good reasons. Events like these enable us to share the work of our amazing academics with the broader audience, stimulating discussion and debate on some of our time's most pressing and interesting topics. And we love talking about what we do and meeting with and engaging curious people like yourselves to share these conversations. As you will hear tonight from our panellists, sourcing opinions and working alongside people from outside of your field can often result in unexpected opportunities. The exciting thing about science is there is always more to discover, and I hope that you will join us again next year when the program resumes to learn more about the latest research discoveries, innovations and impact. I want to touch a little bit on why we are here tonight. As a scientist myself, I have found in my career that the greatest discoveries are rarely made in silos. Drawing from the work of others and collaborating with researchers from different approaches, different technologies and different points of view can sometimes be the difference between making a breakthrough or not. It sometimes happens in very unexpected ways. Our speakers tonight bridge the gap between disciplines every day. They come from different backgrounds. Tonight we have a chemist, an engineer and a physicist. They are each tackling a complex problem to help improve health outcomes for people around the world. These panellists apply their own discipline knowledge to solve medical problems and in new and unique ways. From the future of cancer treatments, enhanced diagnostic imaging for neurological disorders, to the development of bionics that could change the lives of people with spinal cord injuries and other neurological disorders. Each of the panellists is taking a novel, multidisciplinary approach to find solutions. Our moderator will introduce each of the panellists in greater detail shortly, so instead I will focus on her. It takes a special skill to be able to communicate and break down complex science. Tonight's moderator makes it look easy. Dr. Catriona Nguyen Robinson is an immunologist turned science communicator. She completed her PhD in immunology here at the University of Melbourne and the Peter Doherty Institute for Infection and Immunity. Since then, we are lucky to have her in the Faculty of Science as part of our science communication teaching team, guiding the next generation of scientists in the art of communicating complex science. Not only is Catriona an expert communicator, she is also a very active member of the multiple communities she engages with. She wears many hats as the senior editor of Science Victoria, host to her own radio show called Science Queries on Joy 94.9, and the chair of the National Science Week Victorian Coordinating Committee. Through her many roles, Catriona has dedicated herself to sharing science broadly and freely. Many of us will know her as the singing scientist, and you can catch Catriona's breaking it down in more than one way as she creates catchy tunes about science to make it more accessible. I recommend jump on social media, have a look. They're worth a look. Now, while I suspect we won't see one of these catchy tunes tonight, I'm very excited to welcome her and the panel this evening. With anticipation of a great discussion, I hand over to Catriona. Thank you.
Thank you so much, Moira. No, I did not bring my ukulele. Apologies, or you're welcome. You can take that either way. But electrifying medicine. Moira, you said that you anticipate a great discussion. I anticipate a discussion that's going to spark a lot of interesting things. So many different complex problems cannot be solved with one discipline or one field alone, and the human body is certainly complex. So by increasing collaboration between scientists and medical researchers in different fields with clinicians and patients, we can better solve a lot of the significant health challenges by, as what these panelists are doing, generating new diagnostics, devices, and treatments. So tonight we're going to hear how experts from various disciplines are translating their research into better patient outcomes, as well as into healthcare that's more effective, more affordable, and more accessible to everyone. So, to start off, I'm going to firstly introduce the speakers and then I'll bombard them with questions, but then it's over to you. So as we're discussing, start thinking about what sorts of things you would like to hear from the panelists. So we'll have microphones roving in the live audience, and if you're watching online, we'll have someone relaying your questions to us as well. So start thinking. Alrighty. It is my great pleasure to introduce our three panelists. First up, we have Hameson Associate Professor in Physical Biosciences, David Simpson in the School of Physics. David has pioneered the development of high-resolution imaging techniques to better see what is going on in the body. And these advances and this research has led to not one, but two startup companies that David has co-founded, Chromos Laboratories and Phoebe Technologies. And he hopes to use quantum sensing technology, and we'll hear a little bit about what that actually means, to improve diagnosis, treatment, and management of neurological disorders and iron-related disorders. Next up, we have Dr. Catherine Morgan, a postdoctoral fellow in the School of Chemistry. She is designing new personalized radiotherapy treatments for cancer using radioactive metal ions that target tumors, as well as developing diagnostic imaging agents for Alzheimer's disease. And her research uses a combination of synthetic chemistry, radiochemistry, biochemistry, and cell biology. That's already, with one person, a lot of fields coming together. So I'm looking forward to hearing from Catherine, as well as all the panelists on what they do and how they bring these different disciplines together. And last but not least, we have Professor David Graydon. So we've got two Davids. And this David is Clifford Chair of Neural Engineering in the Department of Biomedical Engineering. He is also co-director of the Victorian MedTech Skills and Devices Hub and director of the ARC Training Centre in Cognitive Computing for Medical Technologies. I don't know how you fit it all in. David's main research interests are in understanding how the brain processes information, how to best present information to the brain in terms of bionic devices or medical devices like the bionic ear and the bionic eye, and also how to record information from the brain, such as brain-machine interfaces. He is also conducting research in epileptic seizure prediction and looking at electrical stimulation to prevent or even stop epileptic seizures. So, over to our three panelists. I have just very briefly described your work, but I'm sure you can do it a little bit more justice. So I'm going to give all three of you the same question. I've given the audience a little brief overview of your specialties, but what exactly is the problem or the problems that you're trying to solve, and how are you going about them? Maybe we'll start with you, David.
Thank you. So I'm a quantum physicist, and what that means is I study new materials and try to exploit the quantum properties of those materials to do new things that classical sensors or classical imaging technology cannot do. And so one of the examples that I'll give you tonight where this comes into play is trying to image the electrical activity from individual neurons within the brain. And this is a really challenging problem. Neurons are very small. They're around 1 micron in size. The electrical signals that propagate along these neurons are small. They're microvolts typically. And so we haven't really got technologies to look at individual neurons across a complex network to understand how these complex networks actually function, what are the connection rules and these sorts of things. So I'm trying to develop new materials and new types of sensors that can image this electrical activity with high resolution to provide neuroscientists with the information they need to better understand how these networks function and how drugs impact the function of these sorts of things. And the big problem that we're trying to tackle is around neurological drug development. And so the global burden of neurological disorders is estimated to be around $16 trillion. And Big Pharma is not that incentivized at the moment to trial a whole range of new neurological drug candidates. And so at the moment, there's only around 360 trials going on for these neurological drug disorders. The reason that is because the failure rate for these drugs is upward of 94%. So 94% of these drugs that are trialled fail. And that costs the pharma a huge amount of money and a huge amount of time. And so one of the big problems here is that the models that are used in preclinical trials don't carry through to the human trials. And often the preclinical trials are done on animal models, and then when we want to translate that into human models, they don't work. They're not as effective. And so we want to develop new tools to provide better information and improved efficacy, if you like, of these sorts of systems. And the problem is we're trying to understand this really complex brain, and the way that we do that is we break it down into cells in a dish. And we want to understand what are the electrical pathways of these cells in a dish. And the tools that we have to look at that are very blunt. And so some of the tools that have been developed were the patch clamp. So the patch clamp was developed in the 1990s. It won the Nobel Prize. That was the first time that we could actually look at the electrical activity from a single neuron and understand what was going on. And that's led to tremendous advances in our understanding of these networks. In the 2000s, multi-electrode arrays appeared. As the name suggests, there are multiple electrodes in an array, and that can provide you a certain amount of information, but you can see that it significantly undersamples the complex dynamics of these sorts of networks. And so using these new quantum materials, so my material of interest is diamond, and I look at defects in diamond, we can use these defects in diamond to actually image these electrical signals. And so our sensors are much smaller than these multi-electrode arrays and can sample the individual signals from these individual neurons. And this result happened 2 weeks ago. This is a result that my group has been trying for 7 years to do. This is an example of a single neuron. So here's the cell body of a single neuron. And that little dot is where we're imaging from. So that's one of our probes. We've got probes all over that field of view. And these are the action potentials or the electrical signals that are being produced by that neuron. And so now that we can do this, and we've demonstrated that it works, the biology, we can image the biology, we're now working with a whole range of neuroscientists to really roll this out to understand how drugs impact these cultures and networks. So that's a snapshot of what I do.
Amazing, congratulations, culmination of 7 years worth of work.
Thank you.
Thank you, and now we'll hand over to Catherine.
Hi, so I'm a postdoctoral research fellow in Professor Paul Donnelly's lab over at Bio21, and I'm a research chemist, and more specifically, I'm an inorganic chemist. And what that really means is that I love studying metals. And I like studying metals as well as kind of designing what we can call molecular cages. So these cages that are able to effectively trap the metals within the cage. And we call these metal complexes. And so here is a kind of, day in the life of a research chemist. I've got a reaction going here, and you can see I've got that video is not working. But basically, I've got our white solution here. We've got our molecular cage, and then we can add in that copper. So that's a metal that we might all be familiar with. And almost instantly after I add that copper into our molecular cage, we see a really visible color change. And what that shows us is that we can almost instantly see that the metal is encapsulated within that cage. And so that's really where my research journey started. And it was designing these metal cages, evaluating them, seeing how stable they are, because obviously we need very stable metal complexes if we're going to use them or look at them in biological systems. And that kind of brings me to what I research now, and that's looking at radioactive metal cages for the treatment of cancer. So radioactive metals are very unstable, and when they break down, they emit radiation. And we can actually harness that radiation for a range of different medical research and applications. And what's most exciting to me is that we can harness that radiation to potentially kill cancer cells. And so here's a very candid photo of me looking at my cancer cells. These are prostate cancer cells, and I'm checking to see that they're suitable so that I can do some analysis with our new radioactive metal complexes. And so just very briefly, this last slide, there's a bit going on here, but I think it's a really beautiful slide. It kind of summarizes the kind of problem that I'm trying to solve and what I'm really interested in. And so there's lots of different radioactive metals that exist, but the one that I'm most interested in is this radiometal called actinium-225. It's a very rare radiometal. And actinium, it actually releases very powerful radiation that's able to effectively kill cancer cells. And so really the challenge becomes, how are we able to localize that actinium into the site of the cancer while sparing healthy tissue? And so to solve this, we need to attach our actinium cage onto a drug, such as a peptide or a protein or an antibody, so that it can selectively find and bind that cancer cell. So here is an example of some kidney cancer slides from a mouse model of cancer. And you can see it's a very sensitive technique. Over on the left-hand side, you can see each individual cell. So it's very sensitive. And what I'm kind of most excited by is that we were able to treat our mice that had these kidney cancers with our actinium drug and significantly slow tumour growth. And what's so exciting is that when we look at this kind of green panel here, we can see all these green little dots. Each green dot within that cell actually represents a point of irreversible damage to DNA. And that's kind of the goal, is to have a really targeted radiation therapy.
Thanks, Catherine. I think you said there's a lot going on that slide, but I think it just shows how beautiful science is, like visually, but also like bringing together lots of things to solve problems, as you're doing. And David.
All right, thank you. So I'm the one who works in the bionics area, and so you can see here this the cover of Science magazine way back in 2002 showed what was possible back then. And we can see already that there's a number of organs that have been replaced, knees have been replaced, and so on. So there's these two main aspects of bionics. There's the sort of bionic limbs, joint replacements, that sort of thing. That's not my area. My area is in how we interface with the neural systems. Trained originally in electrical engineering, and so I'm interested in how the electrical system of the body works. And one of the most amazing inventions in prostheses was the bionic ear. And that was first implanted in a person back in 1978 here at the University of Melbourne, and is now the basis of a multinational company, Cochlear, one of Australia's greatest success stories. So that shows amazing success, but there's still a lot of people who don't benefit as much as others, and music doesn't sound very good. So what we're working on now is ways to add different types of stimulation. So we can use light stimulation to stimulate the auditory nerve, and that may allow much more precise targeting of the stimulation because you can steer lasers really well. So another area then that developed out of the bionic ear, also here at the University of Melbourne and with our partners, was the bionic eye. So for people who have lost their vision through retinitis pigmentosa or macular degeneration, it's possible to place this device into the back of the eye to stimulate the remaining cells that exist in the retina. And there we can see that two different locations where they could be. We're going for that location underneath here in Melbourne. But there is also the epiretinal electrode array that's probably a bit further off. It's more difficult. So already 10 people have been implanted with this device. It's undergoing commercialization, but it's very low resolution. So our research now is how can we get the most out of having a very small number of pixels, you could say. How can we combine this electrical stimulation in such a way as to take advantage of the way electric fields interact with each other? Epilepsy was mentioned also, so we're very keen to understand, first of all, why do people have epileptic seizures? It's not really understood, even after centuries of research. But one of the ways to do that is to build systems that we can record people's electrical activity of their brain, their EEG signal, for weeks, months, even years. So there's now a device that's being commercialized that uses these electrodes that are actually under the skin but over the skull, and the first recipient of that has been using it now for 3 years. So we can use these sorts of long-term signals to help us understand maybe not just why seizures occur, but also when they're likely to occur, and come up with this seizure forecaster, much like you have a bushfire forecast where we can have a medium to low risk or extreme risk. We can provide people with this indication of if they're unlikely to have a seizure today or if they're highly likely to have a seizure today, and they can better plan their lives around that. Brain-computer interfaces is going the other way from Bionics. It's implanted devices that can record signals from the brain. So also developed here, University of Melbourne in a fairly large team that is now spun off into a company called Synchron, is the Stentrode brain-computer interface. We can see here a person, there's been about 10 people also with motor neuron disease who've received this device that's implanted within the blood vessels to get into the brain. So we can record from inside the brain without actually having to open up the skull to do that. And you can see the signal is recorded. The person there imagines, say, moving their leg or moving their arm, and that helps them to control a computer screen and do the many things that we can do on computers. That shows the electrodes. We're now looking to see how can we turn that around and place these devices into blood vessels anywhere in the body to stimulate. And so we can stimulate parts of the body without having to do the complicated surgery. So this shows an example of stimulating the pudendal nerve from a nearby blood vessel. So for people with spinal cord injury, it may give them control over their bladder, which is one of their most desired functions. So I think that's it for me.
Some incredible feats there. So each of our 3 speakers are solving medical problems, but all 3 of them kind of come from fields that are outside what we generally consider traditional medicine: engineering, chemistry, physics. So I want to know what you think is the greatest value of interdisciplinary science and investigation to collaborate to improve patient outcomes. Maybe we'll start on this side this time. David?
Right. Well, I definitely couldn't do what I do without collaboration. As I said, I did degrees in electrical engineering and computer science, and now I'm working on devices that you put into people, right? So there has to be interdisciplinary research to do that. So first and foremost, it is through clinicians that we find where the needs are. Some of them really stand out because somebody's blind. Can we help somebody who cannot see anything? But other conditions like Alzheimer's, epilepsy, it's harder for an engineer to sort of understand and see that. And so having this sort of needs-driven research that's coming from clinicians is really important. But then we need a whole team. We need physiologists, we need materials scientists, we need mechanical engineers, electrical engineers, biomedical engineers to sort of bring it all together.
Thank you. Catherine?
Yeah, awesome. So I guess similarly, coming from a chemistry background, I guess if you think of the kind of pipeline from having something sitting on your bench or making something in the lab all the way to actually having a kind of tangible patient outcome, I'm very early in that pipeline. And so, partly some of the things I do in the lab because I'm interested in them and because it's, academically interesting. But then the parts of the research, like, similar to what I showed, that have a potential impact, I am somewhat limited with what I can do. And so to be interdisciplinary is really to give some of your research ideas kind of life or to actually have an application, yeah.
Thanks, Catherine. David?
So interdisciplinary research for me has probably changed the way that do research, I would say. So I started doing or collaborating with people from the medical faculty maybe 10 years ago. And it really changed my outlook of science and the output that we can, or impact that we can have on society. So I used to sit in the lab and play with diamonds and look at different defects, and they have different colours. And you'd write a nice paper on that, and that's great. When I started talking to the medical faculty, there were lots of really important, hard problems that they were trying to solve. And some of the technologies that we were developing could be applied to do those things. And that gave me much more motivation to get involved in those big, complex problems that need different perspectives. And that the perspectives that I've got from the medical colleagues and the neuroscience colleagues has really driven a lot of the new ideas that we've had in the technology that's been developed in our group. So it's a two-way sort of benefit, I would say. So now I'm more driven to do or to have impact and develop new science and new tools that can benefit society in that way.
And obviously this work requires you to sort of go out there and make these collaborations and make these partnerships happen. So David Graydon, I'm going to start with you. You work closely with hospitals in pursuing your research and getting these ideas off the ground. So could you please explain to us a little bit about what it's like bringing these partnerships to life?
It's extremely interesting and also really challenging. So I think one of the first things I learned when starting the epilepsy research. So I was working with Graham Clark who is credited with inventing the multi-channel bionic ear, and he had this idea. Well, can we take the technology of a bionic ear and apply it to epilepsy? Can we use it to stop epileptic seizures? And so he introduced me to Professor Mark Cook at St Vincent's Hospital. And this is the first time I've been interacting with otolaryngologists and audiologists for a while, but hadn't really got straight into a hospital department like that. And one of the key things I found was that we had to learn each other's language. When we were talking to each other, we would like even the word model can mean something completely different between a scientist, a clinician, an engineer. Completely different. And so what we actually did was we met every Friday afternoon and just talked through this for 2 years. And it was amazing. It really opened a lot of doors. So I think that's the way I like to work now across these new technology areas, is to, first of all, try to really spend the time to understand the problem, and also to understand what the person means when they describe that problem, and then not just say, okay, here's your solution, but work together to come up with solutions that will not only look good as an engineering or science solution, but will actually really work in the clinic.
Thank you, David. I think acronyms are a big thing as well. Like an acronym for one person can mean something completely different in another field. I want to hear from David Simpson and Catherine now in terms of your experiences. Like, what makes a good collaborative industry partner or interdisciplinary partner?
So maybe from my perspective, I've been very fortunate in the people that I've worked with, but they've been very open-minded to try new technologies and to try new ideas. And so often the first time you try something won't work. So if they're willing to go again and willing to continue to work with you, that's really important. So I think I've had some real champions in that space that I've worked with over the journey, that have been quite visionary in trying new things and exploring new things. And just getting out there and talking to people. So I don't know how many presentations I've given to medicine. Some I walk out of and they go, who is that crazy guy from physics? We're not getting him back. And then I went over to Flory and gave a talk. So we make really great magnetic sensors as well. So I thought, Flory does a lot of work with iron. Iron's magnetic. So let me go and talk to my people. So I went to Flory and I said, have you got anything magnetic that's interesting to look at? It turned out that one of the associate professors there had a great problem in trying to assess the iron load in our blood, which is done very poorly with the current clinical blood tests. And now we've started this collaboration that's gone on for 4 years. And it started from a random presentation that I gave saying, hey, we make great magnetic sensors, what can we do with them? So we had the solution looking for a problem, and they had the problem looking for the solution. So, but you've got to be willing to get out there and do that, and just be ready to take the criticism when it comes. But you'll find a match. Yep.
Just keep trying. Catherine?
Yeah, I was definitely going to say open-mindedness was definitely something important. I think every kind of different discipline or something has a slightly different point of view as well, which I guess you're kind of, the whole point of science is you're kind of piecing together the picture, like the pieces of the puzzle, and every kind of discipline has something slightly different. And so in all the collaborations that I've kind of had, or different institutions that I've worked with, I've kind of always come away with something slightly different, even if it was something not to do. At least it's something that you're kind of getting from that collaboration.
Awesome. This next question is kind of like your pitch. I mean, you've already kind of given us your scientific pitches, but why is the translation of research into a product or a service that can impact people's lives so important? It's like a little short pitch as to why you think that's important. Catherine, we'll start with you.
Oh, me first. I think, again, it's something I spoke about earlier, was giving life to your research, and without, whether it be industry partners or someone like that, the research kind of can only go so far. And so being able to have a kind of tangible outcome is the main driving force, I guess.
Yeah, see it actually there.
Yeah.
David Simpson?
I think translation is one of those wonderful things that's very difficult to do. So I like the challenge of actually trying to translate something and commercialise something. They don't all succeed. So that's something that drives me from one side of things, but just benefiting people's lives and having an impact, a positive impact on either whether it be their health or management of particular disorders and these things. That's what really drives us to do the work that we do. So yeah.
And everything I do is really about the translation. To me, it's great to do the science. It's great to understand more about the brain and about the nervous system and so on. But it's about the clinical need. And so I think it's an enormous effort to get something from a lab into people, and then an even greater effort to then have being able to produce things at the scale that it's going to help enough people. So that sort of translation is what we aim for, but it is really hard.
And it's amazing that you're putting that effort in, like you and a team. Obviously, is it a big team, but you put the effort in and it gets out there. David Simpson, I have a question for you. Your work is less focused on the sort of patient interface and the patient interaction, but more on helping clinicians in terms of enhancing imaging techniques and diagnostics. Why do we need to be developing enhanced imaging techniques at the moment?
Good question. Because there's a lot of things that we can't see. And if you can't see them, then we can't understand them. And so I'm also part of a new ARC Centre of Excellence in Quantum Biotechnology. And this is a $35 million Centre of Excellence with 5 university partners across Australia. The core mission of that centre is to develop new quantum-based technologies, in this case, that can help inform us and give us more information about how biological systems work. And so at the moment, the state of play for a lot of biological systems is we can take wonderful static pictures of a biological system and understand how things are ordered and arranged. But if you want to understand the dynamics, so how that system changes on the timescales that are relevant for biological systems, that's really hard. There's not a lot of imaging tools or sensing methods to do that. So that's where we see these quantum tools potentially having an advantage for that. In terms of the stuff that we're doing with the neurons and trying to image these electrical activities, the dream there is that we would love, so we don't actually directly deal with patients, but our dream for that technology would be if someone had epilepsy, for example, you could take that person's skin cell, you could convert that skin cell into a stem cell, and the stem cell into a neuron, and that neuron will then form a neuronal network. And that network will actually have your form of epilepsy. And so if we can image all of those electrical activities that happen in that culture, in that dish, which replicates your particular disease, then we could try all of the anti-seizure drugs, for example, for your particular condition of epilepsy, and then recommend which one would be best for you. So that's how we would potentially hope to benefit patients down the track, but that's why we do.
Yeah, thank you. Speaking of benefiting patients, Catherine, you're working on cancer treatment, and cancer is something that's probably close to a lot of people's hearts. Can you talk a little bit more about what a practical application of your research would look like in a clinic and how it might change the way that patients sort of experience cancer treatment?
Yeah, absolutely. I think with this type of targeted radiation therapy, it has the potential to have the biggest impact in kind of cancers that have traditionally had a very poor patient outcome or poor prognosis. So, you know, what we know is that if you have a primary tumor and you catch it early, there's many different types of, you know, surgery or chemotherapy or immunotherapy that can successfully either put you in remission or even cure. It can be curative. But in the cases of metastatic disease, so in a metastatic disease, you have a primary tumor that actually spreads to different organs of the body, and it's typically associated with a much poorer patient prognosis. And so I believe that having a particularly targeted approach has the potential to have the biggest impact, because we not only, like surgery could not potentially be very useful, and so having that as a parallel or something that you can fall back on in the case of a particularly advanced disease, that could be really helpful, yeah.
Yeah, well, hopefully we see that one day. David Graydon, you're applying engineering principles to medical problems, and in your experience, I mean, you're sort of coming in from one of the sides, but what's the difference between taking sort of this engineering approach and applying it to medicine as opposed to taking the sort of more traditional medical approach to these medical problems? It's a big question.
Yeah, it's so well, I think in both cases, we don't necessarily need to fully understand the problem to be able to treat it. So I think that is something that is in common, and that engineers love to do. Like we love to know how you put in an input, what's going to be the output of this system, and then we can trial many different things. With machine learning now and the advances in AI, it's been more and more possible to try out so many possibilities. But I think perhaps one of the key differences is just the mindset of design thinking, and actually they're not really too different. But I think that we really want to understand how a system could be adaptable across people. So David already talked about this sort of personalized medicine approach. How do we build a system where we can easily turn dials and knobs to get it to work well for an individual? And I think that's something medicine is moving towards, but it's something that sort of comes naturally in engineering.
Yeah, I did want to follow up with personalised medicine. So David Simpson, we'll go back to you. Some of the examples that you've given us in terms of what you're working on, that essentially is fitting where we're going in terms of personalised medicine. So could you talk a little bit more about the potential flow and effect of being able to use enhanced imaging and this sort of more personalised approach?
Yeah, I'd preface that with we're just at the beginning of this journey. So this is happening now. There are lots of new companies that are built around this idea of precision medicine, but it's very hard to identify a predictable biomarker that you can act on. So you're looking for a predictable change that replicates a particular disease or a particular condition. So we're not very good at finding those biomarkers yet, and particularly for neurological disorders, that's a really challenging problem. Part of what we're doing is hopefully providing a neuroscientist with more information to make an informed decision or to identify that biomarker. So precision medicine, as far as I understand it, is in its infancy and has a lot of promise, but we'll see whether we can get the right tools to get that information. But I think it will have a profound impact on how we treat people and patient outcomes if we can identify these biomarkers and develop drugs that specifically target or modify their activity.
Yeah, for sure. And all three of you have really touched on personalised medicine. So, Catherine, I might ask you what the idea of moving towards personalised medicine means for you and for your work.
Yeah, so I think in the field of kind of nuclear medicine, even though I'm a kind of radiochemist working in the field of nuclear medicine, it does come back to being able to treat what you can actually see in real time, as well as many other different types of diagnostic techniques, but just in my field, I guess. And something similarly to precision medicine is the notion of theranostics. So it's a very jargony word, but it sums up as diagnostics and then followed by therapy. And so to be able to diagnose what somebody has in real time, to assess the biomarkers, assess what their particular cancer's like, and then adjust the treatment strategy accordingly. And the field of theranostics kind of comes to using the same drug and swapping out the radiometal, and so using a diagnostic radiometal to diagnose, and then once you have all the information that you need, then using a therapeutic radiometal to potentially treat. So that's just one tiny aspect of personalised medicine. But yeah, it's very exciting, but in its infancy.
Yeah. David, did you have any additional thoughts on personalised medicine?
I think I already sort of touched on it. So I think that one of the key areas we're moving into, I mentioned it, especially for the bionic ear, cochlear implants, there are around about 20 to 30% of users don't benefit as very much, so that they can't use the telephone, or they still rely on lip reading, or the noisy environments make it really hard for them to hear. So it's mainly because of advancements in the computing technology, and so far we can create what's called a digital twin, which is sort of like a computer model of somebody where we can try to replicate, say, for bionic ear, how their hearing works. And then we can try different strategies for sound processing and noise reduction and things on the model, which we can then pick the best candidates to try on a person. Because we may have 100 different ideas of how to improve sound for them, but we need them to try each one of them for at least a month. So we can't afford 100 months or the clinical work to be able to work, to be able to work individually with people like every month. So I think that aspect of this sort of concept of digital twin, can we make a copy of an individual so that we can try new things on that model to then try with the individual.
Thank you. You've actually started answering my next question in a way, because and I'm going to lump myself in this, the biomedical research community, or like the larger biomedical research community, we've been excited for personalised medicine for years, but you've said that it's still in its infancy. So what are the challenges, or what are the barriers to actually getting it out there and implemented? David Simpson, we'll start with you.
Yeah, for me, it's finding that technology that delivers that biomarker that we can act on. And so for our work, whether that's an electrical signal that gives us that information that we need, whether it's a genomic signal, proteomic signal, there's a whole range of different things that people are looking at to identify particular characteristics of disease. But it's a really hard and complex problem to solve. So until we find the technology that gives us that answer first, I think then that'll change the game and shift the needle for precision medicine. So stay tuned. I think we're close. Yeah.
A future Science at Melbourne lecture.
Yes.
Catherine?
So what was the question?
The answer. The challenges to getting personalised medicine actually out there on the ground.
Yeah. So yes, very similar type of answer. It's finding that drug, and even looking at the neurological cost and failure rate, taking something, translating it is very, very difficult and has a very poor success rate because it behaves so differently when you get to either the clinic or the different clinical trials. And so apart from obviously cost, it's finding the right new radiopharmaceuticals. Yeah.
David?
And so a lot of what I do relates to the organ level. And so I think that one of the biggest barriers is that we really don't understand how it all works. So the brain, for instance, is so complex. And an enormous amount of science has found like how an individual cell works. There's so much that's now understood. There's still so much that is not understood at that single cell. Imagine now needing to work out how 1 billion cells interact with each other and how there is this, So I think we're just still scratching the surface of understanding how an organ like the brain works. So I think that we can still model this input-output function, which is what engineers like to do, and try to make the most of that. But we sort of need those big basic science breakthroughs as well that will help to build that understanding, and then we can do personalized medicine better.
Yeah, thank you. In a few questions' time, I'm going to be coming to you for questions, so start thinking about what you would like to ask the panelists. You've just spoken about the challenges of personalised medicine specifically, but I wonder what the challenges are for you in terms of implementing your research in general, personalised medicine aside, or maybe that's part of your answer. Catherine?
Yeah, so obviously research doesn't come without challenges and barriers. It makes you very resilient. So I think in terms of the research that I'm doing, I'm working with radiation, and something that's very important to know about radiation is that it is decaying. So even logistically and geography can sometimes be a limiting kind of factor for my research. For example, when I get a shipment of actinium, it's got a half-life of 10 days. So 10 days after I get it, half of it's gone. And we keep kind of repeating that. And in the grand scheme of things, 10 days, you can still get a lot of work done. There are other radiometals that have a 1-hour half-life or a 12-hour half-life. And so you have to plan things very well. Geography as well can be difficult. I think we are pretty heavy hitters in the radiopharmaceutical kind of development at like the Melbourne in general, even Australia. But geographically, we are very isolated, and we're still fairly small in terms of radionuclide production. So there are some isotopes, so some radiometals that they're not produced in Australia. So we have to get them from America and Europe. And so logistically, having all these things come together, my supervisor sometimes says it's like trying to ship an ice cube. So it's melting, and you can see it tracking on the post, and it's taking a while to come there. So it's really complex. It takes a lot of moving parts to come together to get these kind of meaningful results.
Yeah. David Simpson?
I've got the Dean of Science sitting here in front of me, so there's no challenges here in the school.
No, now is your chance to say, like, here are all my challenges.
I think for me, the challenges come in the translation of the science. So I think our group is fortunate to benefit from a lot of funding to do the fundamental science that we do. But when we try to translate, getting funding to do that translation piece, which is often a lot of money that's needed to actually create a product and turn it into a solution that a neuroscientist can use, it takes a lot. And that landscape is changing, I would say, at the university level and also sort of nationally. I think the federal government's looking at different ways to assist in this area. But I think that's one of the major sort of challenges, is how do we navigate that transition from the fundamental research through to a translated or commercialised idea.
Thank you. Yep. David Graydon.
Yeah, very similar there, that the funding, it takes 10 times as much funding so you could do the fundamental research to get a device that could potentially go into humans, 10 times more funding to get it into the first human, and then 10 times more funding again to get it into manufacturing. And so that is really difficult. But also, and this might be touching on another question you're going to ask, there's a strong regulatory environment, and I think it's very important to have the regulations, but this process of going through clinical trials is also quite a challenge.
Yeah, like I said, that's why I asked you last, because the next question is on regulation. But I mean, Catherine, you were even talking about logistics, and I imagine regulation would play into that as well. So I can imagine that the regulation around, getting something from fundamental research into people, into humans, there's a lot of frameworks that you have to sort of navigate in terms of care like this. So what are some of the changes or the new approaches you would like to see taken into account for these regulations around medical care? David,
Yeah.
did you want? Oh,
no, you go.
Me?
yeah,
Or-
go
yeah.
for it.
So as I said, the regulations are very important. We want to make sure that any device that people use is safe and effective. And by safe, it means that there's trade-offs. In any medicine, there's always side effects. There's potential risks with these devices as well. But we have to show that the benefit exceeds the risk by a substantial amount. And so that does take a lot of work. There's a lot of paperwork in it. So, a filing to the Therapeutic Goods Administration in Australia is, maybe you've printed it out, it'll be about that tall. But where it needs to change, though, is because of this rapidly growing area of AI. So currently what you do is you create a device and say, okay, here's my device, this is how it works, this is the program that it runs, let's test it and show that it's safe and effective. But machine learning and AI gives the possibility that this device can learn from the way it's used. It could learn to adapt to an individual. It could learn in all the ways that we're seeing now that AI is being used. But what that does is it's changing the device. Every day the device might be different. And so it's very important now for the regulatory bodies to come to grips with that and to be able to find a way to show that even though these changes are happening, we can say that the device is still safe. Yeah.
Sorry, Catherine.
No,
Go for it.
Okay. Oh, well, just very briefly, I think it is kind of changing. I think in my field, I think maybe radiation has a maybe negative connotation. But I think there's been a couple of FDA-approved medications that contain radiation that have recently, in the last 3 or 4 years, that have been approved. And I think that's kind of maybe socializing the idea that this is actually a really efficient treatment and this can work in cases that have not been successful using other treatments. Yeah.
I try to avoid this by not putting things into people. So a lot of the stuff we do is either diagnosing either a blood sample or in the case of the neurons, it's either derived from a stem cell. So a lot of the regulatory issues, they're not gone, but they're reduced significantly in that case.
Mm-hmm. All right. And my last question for you before we open up the floor is that this is, all three of you, you're talking about like very big and exciting spaces, and I want to know, what are you most excited about in your field to see come into fruition, thinking about the sort of near immediate future? Catherine?
I think, so I'm a fairly early career researcher, and even in the time that since I started my master's or, started my PhD, I've seen research articles that I've enjoyed reading have a clinical translation. And I feel like we're almost at that kind of tipping point where, more than 20 years of research in a similar field of, nuclear medicine is about to potentially have the floodgates be opening and there be lots of clinical implementation of these things. So I'm excited to see what happens.
Yeah. David Simpson?
My goal is to have a quantum sensing technology in the next 5 years that has a positive impact on people's lives, whether it be through improved blood tests or better understanding of neurological disorders.
David?
And mine is to do what we call close the loop. So this is an engineering control principle where we can measure the output of the system and then feed that back to make the system behave the way we want. So at the moment, a bionic ear, it just records sound with a microphone and then it stimulates the nerve, and you can adjust the parameters, but that's just, you set and it's it's what's called open loop. Having systems that are able to record the neural response to that stimulation and then adjust it in real time to optimize it for an individual. So maybe it can record the brain's activity to stimulate to stop someone having a seizure. Maybe we can record the retinal activity to optimize the image that a person sees with a bionic eye. So closing the loop is where it's going.
Well, hopefully we see all three of these things. Does anyone have any questions? Yes, we've got a roving microphone that will come to you. So just wait, please. That way everyone online can hear too.
Thank you. I've got a question for each David. Some time ago, just prior to getting some dental treatment, I asked my dentist how an anaesthetic works, how it numbs the area around the tooth that he's going to drill or whatever. And he explained that the synapses between the nerves were basically deactivated, if that's a good word, and therefore the pain signal did not transmit to the brain. Are synapses the same as neurons?
Oh, a synapse is part of a neuron. So a neuron, so there's three main parts of a neuron. There's the synapses that receive input from other neurons or from receptors and so on, down to the soma, which sort of adds them all together, and then an axon that sends it off to the next lot of neurons. So all neurons are sort of connected together by these synapses. Yeah.
The second question that you showed in one of your slides, David, was related to how you treat epilepsy and how you can sort of help minimise it or even eliminate it by the use of stents. I'm familiar with the use of stents in cardiology. How does it apply to the treatment of epilepsy?
Yeah. So we do know that it is possible to stimulate the different parts of the brain in a way that effectively sort of turns down the excitability. And so if the brain becomes excitable, it's more likely to have a seizure. So there are existing devices already that stimulate in the deep brain structures. They've been very effective for Parkinson's disease. They've been fairly effective for epilepsy as well. So what we are looking at is how to optimize those devices to stop seizures, but also how to be able to put those devices into the brain without having to drill through somebody's skull to do so. So the stent technology is a way of actually using the blood vessel highway as a way to get into the brain, much like how stents can be put into a heart, into a coronary blood vessel via a vessel in the groin, right? So it's become so much easier to do stenting in the heart because of these technologies that exist. So we're looking to see if we can do the similar in the brain.
And did you have a question for David Simpson as well?
No, in fact, I thought David Simpson raised the point of neurons, and I wondered about the similarity to synapses. But just on stents, so with the heart, it's obvious that the muck collects in your coronary arteries, and they put a stent in to just widen it and create a bit more space for the blood to flow through. So is there a similar process in applications for epilepsy, for treating?
Oh, yeah. So I suppose I haven't really gone into much detail there, but what we've done is to take a stent that would be used for opening up a blood vessel, but placed electrodes on that. So there's electrodes mounted on the stent and wires that then come through the blood vessels. So, for instance, for the brain-computer interface, which is where it is being used in humans now, you can actually place it through the jugular vein up to the top, this big blood vessel at the top of the head that sort of sits in the area of the brain where you control your muscles. And then a wire comes down out of the jugular vein into an implanted device that can wirelessly transmit the signal to the outside world. So the stent is really just a carrier for those electrodes and a way to be able to put them into the blood vessel, and they can be there for the rest of a person's life.
Do we have any questions for all panelists, or Catherine and David Simpson? Yes, at the back.
I have a question for David Graydon. David, I have a friend who has macular degeneration in her left eye, and she gets periodic injections to stave off the degeneration. But you spoke about the bionic eye, and I don't know if this is related to this, but is there something that can be done about that bionically, so to speak?
So the bionic eye is only going to be used for people who've completely lost their vision already. So it's not. There may be potential for bionics approaches to help regenerate nerves. Certainly, there's research in the Department of Otolaryngology here and the Department of Audiology looking at how to use electrical stimulation to promote the auditory nerve to grow back after it degenerates. So potentially there is that possibility, but that is really cutting edge at the moment.
We're going to mix up things a little bit. We will come to you, but we're going to go online first.
We have a question for the entire panel. Can any of you talk about the international regulatory landscapes in implementing solutions like these? In your experience, are Australia's regulations limiting or encouraging innovations in these areas?
All right. So maybe, if you don't mind starting, Catherine, we'll start with you just because you were talking about the fact that you are sourcing things from overseas and things. So I wonder how international regulations sort of impact your work.
Yeah, so there are a lot of regulations. I find also working with different institutes, everyone also has their own regulations. Then there's the Australian government, and then there's the Victorian government. They have separate regulations, and I also work in Queensland, so there's also the Queensland government. So they all have slightly different regulations, and I guess it's just working out the best way to kind of still perform your research in a way that you can kind of cut through all of the paperwork. But in terms of FDA approval, TGA approval, I don't think I can really speak to the regulations, yeah.
Fair enough. That's why you work in a team, right?
Yeah.
David Simpson?
Again, I'm not an expert on the regulatory pathways. The conversations I've had with people, in terms of whether it helps innovation in the country, the pathway I've been told is to go and get FDA approval first, and then the TGA will approve it. So whether that is creating innovation in Australia, I'm not sure, but that's been what I've heard on the grapevine. But maybe David.
Anecdotal.
Yeah, anecdotal. So maybe David has much more experience in this space.
Yes. So actually, there's two types of regulation, and I think both of them are actually really good. So one is around ethics. We want to make sure that our research is done ethically and to a high standard. And the National Health and Medical Research Council oversees that, and they do an excellent job. And this university has multiple ethics committees. Each hospital has an ethics committee. On the other side is that translation regulation, and yeah, it's each country has its own regulatory body, and each country needs to approve the use of devices in their own country. However, there's a lot of agreement about what should be regulated and what shouldn't, and understanding there. So the FDA, sure, is often the one to go for first because it's an enormous country. The United States is an enormous country. They have a lot of resources and a lot more capacity to do the evaluations. And then the TGA can see that and say, well, you got through FDA, you still need to show to us what you have done, but we understand that the FDA is rigorous enough that we would accept that. Similarly with the European regulations. But it's not, so the regulations is hard, but it's not bad, right? It's so important that we have these regulations.
Was there another online question that you wanted to ask before we move over here?
Yes. So, David Graydon, you touched on it, but there has been lots of discussions about generative AI and machine learning recently. For each of our panelists, what do you see as the greatest opportunities with these technologies and possibly the biggest pitfalls to your research?
David Simpson, do you have an answer? Do you want to kick us off?
In terms of machine learning and AI, the way that we're looking to interface with that is we're going to rapidly produce vast amount of data where in that data we want to identify this biomarker that I've been talking about. So how do we look at all of those electrical signals that come from this really complex network and then understand which patterns are important? And so machine learning is really good for that. So we're looking to use it in a positive way to pull out predictive biomarkers. So that's how we would use it.
Yeah. Were there any pitfalls? Sort of all, it's all good.
It hasn't given me the answer yet, so I'll come back to you on that one.
Yeah, fair enough. Catherine?
I think it does have the opportunity in the design of, bioactive agents or drugs, or especially biologically based compounds. So there's programs like AlphaFold, which has, revolutionized the way that people look at proteins and predict, three-dimensional structures. And so it's very exciting. And again, there's so much data that we almost don't even know what to do with it all and how to utilize it correctly. I think maybe the only pitfall would be that sometimes it doesn't work. So it can be an excellent prediction and it can give you a lot of information. And I've had experiences where I've been told that this peptide, this is going to bind to the epidermal growth factor receptor. And then I've made these peptides and unfortunately they do not bind to the epidermal growth factor receptor, even though theoretically they should. And so I guess just taking that knowledge to supplement our understanding, not to replace what we are doing as researchers.
Yeah. David, you did sort of mention it with the digital twin, but do you have any thoughts?
So much of what we do relies on machine learning. So just making sense of that large amount of data, we record a brain signal to try to control a cursor on a screen or a robot arm, and it's for a person to sort of sit there and try to work out rules to do that is just impossible. So if we can feed the data into machine learning algorithm, and generative AI is now really looking and holding promise for this, that it can make things possible that are just impossible without it.
Did you still have your question?
You said that you had a solution looking for a problem and you happened to speak to someone who had a problem looking for a solution. Is this finding each other happenstance, or is there a more formal way, or should a more formal way be set up, of merging and meeting the problems and solutions between faculties and departments?
That's a really great question. I think, so the university does try to help foster this interaction. So there are a number of cross-disciplinary institutes that have been established. And really the sole purpose of those institutes is really to start to connect people from different schools and different faculties to just have a conversation about what they do, what are some interesting things that they're working on, what are the hard problems that they're trying to tackle. I've found it's been organic. In all of the times that it's really worked well for me, it's been organic and it's been through happenstance. So I feel like it's very hard to engineer sometimes, but you never know who you're going to meet at particular venues. So I had another example of a collaboration. I went to an engineering event, never been to one in my life. I thought, okay, I was asked to give a talk there and didn't know anyone in the room. I had a great conversation with a guy over coffee, and again, we've been collaborating for the last 5 years. Would never have happened if I didn't go to those things. So you don't know when it's going to happen, or I think it's very hard to say, yes, I'm going to get 6 collaborations out of this event, but you need to make sure that the events happen. And I think the university is trying to do more of that and recognises that interdisciplinary research really has value and is needed. Yeah.
David?
Yeah, I agree with that. There's I think the university has been really good at creating these opportunities for that serendipity, or for people to get together through whether it's through formal institutes that run seed funding programs or just networking events, through to just trying to make sure that our seminars are well attended across different departments and different faculties, because we're essentially getting like-minded people together, and it's very hard to direct research from above and to say, you know, we have to focus on this, because there are so many ideas that you don't think of until the right people come together.
Catherine, I wonder if it's slightly different for you. You mentioned that you're earlier in your career, and I wonder how that impacts, finding these collaborators.
Yeah, so our lab's situated it's called Bio21. So it's a molecular biology, biotechnology. So it is one of those kind of institutes that has chemists and biochemists and people from pharmacology and stuff. So you are surrounded by very different types of researchers, and I do think that potentially does maybe rub off on me. We've, our lab next door neighbours, they're organic natural product synthesis. They're, the hardcore organic chemists. And there's, been examples of we make these metal compounds, these metal cages, and they potentially use them for different types of catalysis. And I guess in that respect, it does work really well. But I think if I had just offered this compound for no reason, it might not have happened. It's only when there's a problem that you kind of want to help solve it, I guess, in my opinion.
Thanks. Is there another question sort of on this side? Back to the middle.
Question for Catherine. 2 questions, actually. Can you refresh me on the atomic number of actinium?
So it's actinium 225. So it's right down in the actinide series. So if you've got the big periodic table,
Yep.
And then there's the kind of forgotten elements down the bottom, and it's right on the bottom. And so it's it's hiding down there. And it actually, if in case you haven't heard of it, it doesn't really exist in non-radioactive form. So there's no non-radioactive isotopes of actinium. There's some that have been kind of naturally identified, but then most are synthetically produced.
Okay, because I'm thinking what I used to work with in my BSc days was uranium 92-235.
Yeah, so it's
and
a few.
so actinium is 225, but what's the atomic number?
90.
Have to be high 80s or something?
Yeah, I think it's like, sorry.
Anyway, I was just
Not all scientists know everything.
The real question, Catherine, you talked about radioactive metal cages for cancer treatment.
Mm-hmm.
And what I've deduced from reading the newspapers is that when somebody has a cancerous prostate or liver or whatever, they get something that looks like a laser gun and go bzzzt,
Yeah.
and treat the area with radiation. What are metal cages? What are these radioactive cages?
So I guess the thing that you're referring to is called external beam radiation therapy, and that's actually quite common. In terms of thinking about how many people get cancer treatment in Australia, maybe 40% receive external beam radiation therapy, where they shoot that laser beam at you. But that only really works on surface level cancers, primary cancers, so the original cancer. It doesn't necessarily work as well when there's diffuse disease, because it needs to be so precise to shoot that external beam that within, the really complex biological system below in the organs, it's not really feasible. And so the research that I'm talking about is called systemic radiation therapy. And that's when you create this radiopharmaceutical, so a drug. And so we have our metal, and then we have our cage, and then we attach that cage to basically a peptide. So it's very popular, not very popular. It has been used for prostate cancer, and that is injected into the patient. It accumulates at the site of the tumour, so it travels all throughout your blood, and it would accumulate at the site of the tumour. And so that allows for, if there was metastatic disease, it could accumulate at each and every tumour if it is selective enough. So yeah, there is slight difference, yeah.
Thank you. And we're jumping back online.
Amazing. We have a couple of questions from online. The first is about Bionics. So David Graydon, you talked about music not sounding very good with bionic ears. What are some of the quality of life additions you think are important for researchers to consider when developing bionics?
Yeah. So it's always been understood or believed that communication is one of the most important things for people who have lost their hearing, and fortunately, I think the reason the bionic ear has worked so well is that the speech signal is quite redundant, and there aren't the frequency components of what you're listening to at the moment as I speak are fairly broad and not particularly specific. It becomes more important to get those frequencies exactly right when there's a lot of background noise, because you need to distinguish my voice from perhaps other people's voices. So people with bionic ears can struggle with that, because and the reason for that is that in our normal hearing, in our inner ear, we've got about 3,000 different frequency receptors, each a different frequency. So we can distinguish that many frequencies when we're born. And as we age, we start to lose them. But compare that to the bionic ear has 22 electrodes. So only 22 different frequencies to represent it's called the bottle. It was called the electroneural bottleneck, and it's trying to take the rich world of sound into this amazing brain that can handle so many frequencies, but through this tiny little channel where there's only 22. So for speech, that's fine, because we could probably get away with 6 or 7 for speech. But for music, and trying to distinguish the harmonics and the harmonies and that complexity, it just can't represent them very well. So it has always been a consideration, is how can we create that greater complexity, but it's just hard to do. And so we keep working at it, but it's something maybe we'll achieve in the future, but is just not possible now.
Keep chipping away at it. Were there more questions?
Yeah, so we have a question from school leavers here. Do you have any advice for school leavers who may be considering medicine on alternative pathways into solving health problems like you all are?
Is anyone ready to jump in?
Oh, I could jump in. I think stay curious. So part of my teaching here at the university, I teach first-year biomed students. So they're very firmly focused on medicine as a career. And once they have to do physics, or they did have to do physics, and once they get into it, the curiosity that they have amazed me and changed the direction for a number, not all of those students, but some of those students, like just trying to understand how systems work at a deeper level, sparked a new curiosity. So I think, yeah, my advice to school leavers is to always pursue what you're passionate about. I think you'll do well in that area. So stay curious and enjoy physics. It's my plug.
A little plug for physics in there too. Yeah. Catherine?
As someone that maybe, I think I finished high school more than 10 years ago. But I do remember having really no idea what I wanted to do. I knew I wanted to study something, I just wasn't sure. And so really just start somewhere. If you don't like it, you can always change. I started an arts degree. And I decided it wasn't for me, and then I kind of fell into chemistry and fell in love with it. So don't be afraid to try something, and it's okay if you don't like it.
Totally.
Yeah, I agree with that. And I think that in this, just what we've been talking about is interdisciplinary. We really need people from many disciplines. So pursue the area that really interests you. And if you're aiming into something like medical technology, there'll be a place for you, because we need everybody, all the different backgrounds. But there's so many other things. Like I started out, I was expecting, even when I was doing my PhD, to work in telecommunications. I was going to get a job at Telstra.
Me too.
Yeah, and then I got this phone call from Graham Clark at the Bionic Ear Institute saying, we need a speech processing expert. And I jumped at that and completely changed my life. You know, to switch from that, I just love technology, to really seeing how technology could make a difference for people.
Yeah, echo all the points. We had a question up there.
How far is stem cell research therapy still? I know it's been used, and it has had a lot of benefits in recreating new cells and healing. How's that going? Is that still being used in a different format or way, or is that not part of the emerging tech fields anymore?
So I'm happy to take that one. So what I'm talking about is a little bit different to that. So we're not implanting stem cells into a person to promote some sort of treatment. I think we're taking advantage of all of that technology and learning how to deal with stem cells, how to differentiate stem cells into the type of cells that we want to work with. There are people at the Alfred, at the Florey, and companies starting now that are using these stem cells and creating these networks. And it's really at the point where they're trying to understand, how do we use these networks to get the information that we need? So it's not, I would say it's not being used yet in the way that stem cell therapies are to treat other conditions, particularly for the neurological side of things. But we're using those same tools. And so there's a lot of people creating these epileptic models and using these multi-electrode arrays to understand what the signals are and how do we work with that information. We want to jump into that workflow, give them more information, and then see where that takes us. I'm not sure if that answers your question.
No, you've covered most places.
Yeah, okay.
It's an evolving...
Yeah, very much so.
I know it's been used for a long time, I think, in the Germany and in the States.
Yep.
They're way advanced, and they've been used also for people who've had brain dysfunction.
Yep.
Besides, people when they got bruised knees or rebuilding, but they're more aiming at that area because they know that the synapses sometimes is broken down in the communication. But then if you can go back to the brain plasticity, when you got the duplicate mind, so they're working on that. I think there was a research, I think it's gone back, it was in Germany or one of those countries that had done a lot of things, and they're still going to the next stage. That was the only question I asked. Thank you very much for your time.
Yeah,
Thank
no
you.
Welcome.
We've got a question here. The person behind you was first, and then we'll...
Hi, thank you for sharing your insights. Certainly very interesting research. So a big part of research translation is collaboration. So my question to the panelists is, how did you navigate during those early stages that science communication between different departments or different healthcare sectors or even the business side? What were the challenges, or do you have any tips and tricks to navigate effectively communicating that science into research translation?
I love it. Science communication is my jam. David, you started mentioning, well, you sort of talked about the beer story of like having like drinks with clinicians. Do you want to kick us off?
Yeah, so I think that one of the key things is learning each other's language, as I mentioned, and it's it sort of it's very important for each person to recognize that this other person, I'm not going to achieve what I would really like to achieve without collaborating with these other people. And I think that having that mindset really helps to build those collaborations up. But then as you go along, you find that there's these gaps. And you mentioned the business side. Yeah. So that's one of the last gaps that I learned of, because when I was a junior researcher, it was just great getting things done. Other people dealt with that. But as you move more into leading it, you realize how much that translation is going to depend also on people who can do the business side of things.
Thank you. Catherine?
Yeah, I think in terms of collaboration, it does kind of boil down to having clear communication, obviously, between the collaborators, but also maybe an understanding of what do we want to what are the tangible outcomes and what's the timeline, and making sure we're all on the same page is incredibly important. And what are the kind of, I guess, in terms of business, there will be milestones, so that does kind of break it down for you in a way, because you do have these kind of deliverables. But yeah, I think as long as you can clearly communicate and express what you need and what you expect from this relationship, because it is always a two-way relationship, you're each getting something from each other, that's probably the best way to go about it.
David?
So collaborations, the things that help foster collaboration are coffee and beer and lots of conversation. So I think for institutions that want to do good interdisciplinary research, it takes time. So it takes time to learn that language. And so researchers and scientists need to be given that time to actually have a genuine conversation where both parties learn. And that's one of the funnest parts of interdisciplinary collaboration. You can be dumb. You can sit there and have, not that I do this, Moira, but we sit down and we have coffee, and if you've got an interesting question about biology, you get a great biologist across from you. It's not a textbook, but you can just have this wonderful conversation and learn something new about a completely new system that's outside your field, and it's fascinating. And it just gets addictive. And so then it's just, as long as you can have that conversation openly and ask lots of stupid questions and not be judged in that way, it's fantastic. So I think just lots of communication and giving that time for that relationship to really build. And I think Catherine touched on it as well, having expectations that are clear. So we're not all going to create this amazing result straight away. It's going to build. Let's start off simply and then build up from there.
We might just take one more question, and I think we had two. Did you still have one, or go for it?
The gentleman there sort of preempted the first part of my question. You talked about taking skin cells, turning them into stem cells, then turning them into neurons, and then building up the neurons and doing experiments on them. Have they been able to do that yet? Take a stem cell, a skin cell, turn it into a stem cell, and turn it into a neuron?
Yep, so absolutely. So that has been shown to be done, and there's been electrical recordings from patients that have epilepsy and then taken through that journey. One thing I haven't mentioned is how complex that network is. So these networks aren't just neuronal cells. There are other cells that are in the brain that help in the whole communication and signalling pathway. So there are glial cells, astrocytes. And so there's been a change probably over the last decade from looking at these two-dimensional cultures, which I showed images of above, to a three-dimensional model where we've got a mixture of all of these cells together, which is more replicating what's happening with the brain. So there's a startup company called Tessara that are trying to develop these brains in a dish, if you like, that replicate disease in that way. But that's more of a three-dimensional culture that's got a mixture of all of these different cells that you need to better replicate what actually happens with the brain in terms of cell mechanics and these sorts of things. So it's been done in the two-dimensional model. When you move to the three-dimensional model, it becomes much harder then. Then how do we get the signals out of that three-dimensional blob? So we're working on electrodes that work in 3 dimensions that can work on these brain in a dish models.
The other part of my question was, could you please tell this non-physicist, what do you mean by a quantum signature?
It's not I wouldn't say it's a quantum signature in this case, so we can have a separate discussion in the break. The technology that I'm talking about here doesn't use the quantum properties of the materials. When I talk about quantum properties of materials, it's about either putting that system into a state that you can't do classically. So the famous example of that is we play with electrons within these defects. And we can manipulate these electrons into whatever state we like. We can manipulate it into an up state or a down state. And in quantum mechanics, it's possible that if we know how long to manipulate into an up state or a down state, we can manipulate it into exactly a superposition between these two states, where it's in both states at the same time. And you can take advantage of that to do interesting things that you can't do classically. In a classical system, it's only up or down. In a quantum system, it's both at the same time.
I did promise it would be explained, and so now look, we've slipped it in there right at the end. I want to end on like a positive note. So 3 words. What do you love about what you do? They didn't know that this was coming. Is anyone ready to jump in with their 3 words? David, you look like you're...
There's so many.
10 words or less.
Well, collaboration is one. Learning is another thing I love about what we do, and having impact is something I want to do as well.
With hyphen in there, yeah.
Yep.
That's hard. I would have to say exploration for exploration's sake, and then translation, and I'll have to think on that 3rd word.
Well, perhaps it's very broad, but solving unmet needs.
Love it. Thank you very much. Thank you, everyone, for coming along and for your questions. Yes, please do join me in thanking our panelists. You're brilliant. So thank you to you who've come in person. Thank you to the people online. Thanks for also sending in your questions. Thank you to Professor Moira O'Brien for hosting the Science at Melbourne Lectures, and hopefully we'll see you in 2025. And thank you to Taylor, Holly and Madison, who have been behind the scenes, and the tech team as well. But yeah, stick around for refreshments. You can unpack the brains, the ideas, the concepts a little bit more. There are refreshments just outside, so stick around. Otherwise, enjoy your evening, and please, again, help me in thanking Associate Professor David Simpson, Dr. Catherine Morgan, and Professor David Graydon.